My topic is HiPSC as a model for human atrium. I think HiPSC cardiomyocytes have entered the arena, and there's a lot of work ongoing on tissue repair, on target identification, safety pharmacology, and there's a lot also about individualized medicine, mostly focusing on rare mutations in ventricular arrhythmias. Situation is different regarding the atrial iPSC-derived cardiomyocytes because at present there is no clear need for repair, for tissue engineering. There is no need to do safety pharmacology so far for the atrium, maybe for target identification. But I think there's a lot of space for work focusing on single nucleotide polymorphism of genetic variants of unknown significance, different disease states, comorbidity, and so on. So the first question is, are there really atrial iPSC cardiomyocytes? And I think the idea that atrial iPSC-derived cardiomyocytes exist originates even from the very first paper, that's from Ma in American Journal of Physiology. And what people did, they measured action potentials by sharp microelectrodes and grouped microelectrode measurements in 3 groups. And they more or less arbitrarily divided them in 3 groups based on differences in action potential at 90% repolarization. And they called the short ones, given here, atrial, whereas the long ones were defined by authors as long. It took just 4 years that the question that this assumption was criticized by a very elegant paper in Biophysical Journal, where people have used voltage-sensitive dyes, and this technique allows to measure a large number of cells. And these are the original action potential recordings. And this is a frequency distribution of action potential duration at 90% repolarization. And what becomes clear from this study is that the population is rather homogeneous in the culture of iPSC-derived cardiomyocytes. I have to say, I'm very happy that I got the invitation to a NANION symposium because we are working on the other end of the technological development. We use a very low true output technology. We fabricate intact EHT and measure by sharp microelectrodes action potentials from those constructs containing about 1 million iPSC-derived cardiomyocytes. And when we started our work, we first asked ourselves, how can we discriminate between ventricular and atrial tissue, human heart? And here you can see 2 typical examples of a atrial action potential and the ventricular action potential. And this is an action potential recorded in such a engineered heart tissue. And first we tried to replicate the findings in the Ma paper and plotted APD90 values as a histogram. And in fact, right atrial tissue shows a little bit shorter APD90 compared to the green left ventricular, but the difference is very small and there's a huge overlap. The same holds true for resting membrane potential. You can read in almost every textbook of physiology that the resting membrane potential is more negative in ventricular compared to atrial tissue, but again, difference is small, there's a huge overlap. The only parameter that allows a clear differentiation between atrial and ventricular tissue are parameters that reflect early repolarization, and it's very popular to calculate so-called repolarization fraction, where APD90 or APD90 minus APD50 is divided by APD90. Thus, we would conclude that we have low power parameters to discriminate between atrial and ventricular, and the only stable, sufficient parameter is repolarization fraction. And second conclusion was there is a rather uniform ventricular phenotype in traditional iPSC cardiomyocyte cultures. Then we saw that paper by a Dutch group that describes that the exposure to retinoic acid just for 3 days gives an atrial phenotype. And we were quite impressed of the original recording shown you on the left-hand side, because it resembles more or less closely the situation in human atrium. Again, we were even more impressed that some compounds used by us as blocker of atrial selective potassium currents showed nice effects in the atrial population compared to the ventricular population. Therefore, we sent Marta Lemme to the Netherlands to learn this technology. And you can see here on the right-hand side, Marta's results, no effect of low concentrations of 4-aminopyridine that blocks the atrial selective IKUR, whereas there was a clear effect in the retinoic acid-treated EHT. But there was one small issue, because the effect shape differed clearly from those in human atrial tissue, where the response to block of IKUR was a shortening, not as in Marta's results and in the Dutch results, a prolongation. Okay, but we were quite happy to have a model and to work. Marta was I think he was tired from all those recordings and moved to Nanyon. And another young lady entered the arena, and she came to me and said, Torsten, something has happened, really unexpected. And she showed me such original recording. I said, okay, it looks fantastic. It looks like real human atrium. I said, what have you done? They said, no, anything nice. I did anything correctly. And after a while, I got aware that she was very short in time by culturing the cells, and she told me that she has left one step in preparation of retinoic acid, sterile filtration. And then we got aware that many of the filters that were used in cell culture can absorb huge amounts of retinoic acid. And at that time, we had a publication in Revision, and they asked us, okay, what is the reason why you have now such nice action potential shapes, atrial-like, compared to your previous results published by Marta Lemme? And we said, okay, it may be because of the filter. And I said, okay, if you believe that it's really the case, then you have to make a complete concentration response curve on ion channel activity. And these are some original traces recorded in iPSC-derived cardiomyocytes. These are potassium outward currents elicited by test pulses to plus 50 millivolt. And we have exposed cells subsequently to two different concentrations of 4-aminopyridine, a low concentration that blocks atrial selective IKUR, and a high concentration that blocks ATO expressed both in atrium and in ventricle. And you can see, first glance, you can see there is a huge increase in potassium outward currents in general, and the amount, the extent of ion current sensitive to low concentrations of 4-aminopyridine get larger with higher retinoic acid concentration. These figures summarize the results, and what you can see is even with a very low concentration, you have always an effect of the high concentration. It indicates presence of ITO, and only high concentrations of retinoic acid are sufficient to evoke an IKUR-like current. We measured action potentials, and the originals are here shown. And I show this figure because I think it's really important to make clear that the effect of retinoic acid on atrial differentiation is not easily black and white. Even with low concentrations, you see an increase in beating rate. you see a shortening of APD20, but you need very high concentrations, 1 micromolar retinoic acid, to get APD20 values as short as in human atrium. And I say these are high concentrations because 10 or even 3 micromolar will kill your cultures. Then we measure the effects of blocking IKUR on action potentials. And again, it's not black and white. As in the paper of De Valla, as in the paper of Marta, even low concentration of retinoic acid can evoke an increase in APD20. But the APD90 is prolonged in iPSC-derived cardiomyocytes treated with low concentrations. Again, you need very high concentrations to have the action potential shortening. And when I talked this story, I was asked sometime, okay, why have you spent so much time on this specific topic? Then I would come to an older paper from 2004, where at that time in Dresden with Herbert Himmel, we did some measurements in action potential from human atrium, and we blocked with 4-aminopyridine the IKUR current, and what we saw was that unexpected shortening of action potential duration at 90%. This was completely unexpected, and the reviewers asked, okay, how can you explain it? And we said, maybe it's a secondary activation of IKUR, a stronger activation of IKR as a result. And then I said, okay, if you think it's so, then you have to show this. And this is here given for experiments where first IKR was blocked by E4031, and on top we block IKUR. And then we see a shortening of APD90. And why is that point so important? Because the finding, the apparently paradox finding of shortening of APD90 by IKUR block was confirmed with several highly sensitive IKUR blockers. And I strongly believe that this action potential shortening upon IKUR block may, at least in part, explain the failure of IKUR blockers in clinical trials. Thus, I would like to conclude this point that atrial EHT as a pharmacological model critically depends on how closely such EHT resemble native human atrial electrophysiology. What about other parameters to identify an electrophysiological phenotype that resembles the atrium? There is IKACH, and this is again an older publication of us where we exposed cells, iPSC-derived cardiomyocytes, to to carbachol. And this here on the left lower part is an example of a human atrial cardiomyocytes, and I'm happy to see Niels in the auditorium because I think he did such experiments more than several thousand times. What you can see here is that in the normal, not retinoic acid-treated iPSC-derived cardiomyocytes, we never saw a response to carbachol. But in the retinoic acid-treated, here shown on the right-hand side, there's a clear effect of carbachol and I think it's important, in every cell, indicating that at one time the not retinoic acid treated seems to be a homogeneous ventricular population, whereas the retinoic acid treated seems to be an homogeneous atrial population. Again, to make it clear, it's not black and white. Even with low concentration of retinoic acid, you can see small effects of carbachol on APD90, on takeoff potential, as well as on beating rate. But again, large effects can be seen only if you treat cells with 1 micromolar. We have sent that paper, that manuscript, to Stem Cell Reports, and I have to say it's a very solid journal, and they ask us, okay, can you reproduce your findings? And I think this is some of the dark side of iPSC cardiomyocytes, because they forced us to do three different differentiation without retinoic acid and with retinoic acid, to repeat this in two different cell lines. And I think what the figure shows, the individual data points, every point represents one single EHT, that there is a very nice reproducibility in one lab. However, if you screen the literature for action potential shapes in iPSC engineered heart tissues, treated or cultured from cells, treated with retinoic acid, you will see there is a huge variation in APD20 values. So I think the reproducibility of retinoic acid treated is an unsolved issue between different labs. Now you could say, okay, what could be a nice application for such a atrial model? And we made a next step and focused on single nucleotide polymorphism. And there's one single nucleotide polymorphism that is highly associated with atrial fibrillation. It's on chromosome number 4. And look, the y-axis is split. This is the highest association that can be found for a single nucleotide polymorphism in electrophysiology. And then we said, okay, let's knock out that region. That's a region close to PTX2. It's not PTX2, it's close to PTX2, to a transcription factor. And we did it by CRISPR-Cas9. This can be done nicely in iPSC-derived cardiomyocytes. Then we started to measure force development in such atrial EHT, and we saw a smaller force in the PTX2 knockout, and this situation resembled, as we found, nicely the situation in human chronic atrial fibrillation, where contraction forces are reduced. And this is paralleled by a decrease in calcium current measured in isolated iPSC-derived cardiomyocytes. Again, action potential shape differed widely. We had a much longer APD20 compared to the isogenic control, and this relates to a drastic reduction in transient outward currents compared to isogenic control iPSC-derived cardiomyocytes. And again, this situation resembles the findings in chronic atrial fibrillation. So we found that knockout of PTX2 can at least induce clear electrophysiological changes, but we should be aware that the situation of PTX2 knockout is far from any physiology and also far from any pathophysiology. So I would like to summarize our findings and would say the early reprioritization is sufficient or is powerful to discriminate between atrial and ventricular action potential phenotype. The EHT can recapitulate atrial pharmacology. I showed some data on IKUR, IKACH, but there's also a huge difference in serotonin-induced inotropy. The EHT can be scaled up in size and structure up to so-called giant EHTs with several centimeters long. They can be easily genetically edited, and reproducibility may remain an issue. And if I would give you one take-home message, if you start to work with atrial EHT, you should never filtrate retinoic acid. Here are some people that did the work. I would specifically also call Marta and Andras. They, at least Andras, spent some time at Nanion. Marta is still there. And of course, we need always the direct comparison to human heart tissue, and therefore it's for us, it's essential to have a very good contact to cardiac surgeons. And I thank Thomas Eschenhagen and Arne Hansen for production of the iPSC-derived cardiomyocytes. Thank you very much for your attention.
Thank you so much, Torsten. This was really, really exciting. And it's nice how some maybe errors can give such big impacts into experimental flows. We have some questions here coming all around, so I will look up and down and left and right. How old are the HTs at the time of sharp electrode measurements? And is there variation in the same tissue if it gets older?
Okay, we always use 4 weeks. I think this is a nice compromise because every week it's you have to spend a lot of money to take them in culture. You have a higher risk to get infection. And there is a change over time. There is a decrease in beating rate. The decrease in beating rate over time is not so pronounced as we see it in the ventricular, but there is still some reduction in beating rate. I'm not aware about specific differences regarding ion channel blocker response or something like this.
Thank you. And then we have from, as I can see, I think this is Claudia Altomare. So she's thanking for your talk, and she's asking, so we know that the use of the dynamic clamp technique is useful to improve the availability and functionality of voltage-dependent ion channels, which are often downregulated or not fully active in iPSC-derived cardiomyocytes. Her question is then, how do you think what do you think about the use of the dynamic clamp for testing of ion channel blockers, and if it can be an advantage or not?
I think one critical issue with when you work with iPSC-derived cardiomyocytes is their size. I think they are very small compared to adult cardiomyocytes. They are almost 10-fold smaller. And we are afraid that one issue could be an improper and problematic relationship between the current that is flowing through the membrane compared to the leak current. So I'm not 100% convinced that dynamic clamping can solve that issue, but we do not have any own experience. And maybe it's because I'm in a quite comfortable situation. We produce a lot of iPSC-derived cardiomyocytes, and for us, there is no need to leave that very old technique.
From Marta, a question about the PTX2 knockout EHTs.
Yeah.
Do they show any remodeling sign of atrial fibrillation, like fibrosis, for example?
No, I think these are pure, these are EHT containing only iPSC-derived cardiomyocytes. There are no fibroblasts in. And to be honest, I think it was a nice paper. I think it was for us to convince that there is something really going on with PTX2 or genes close. But I would say that the situation is so far from pathophysiology that I'm not 100% convinced if it's worth to follow PTX2 knockout so intensively. I think there are a lot of different gene variants known close to PTX2. There are so many other gene variants that could be studied using atrial iPSC-derived cardiomyocyte models. Yeah.
And maybe the last one, we've seen all of this different variation of this atrial action potential across different labs. What is your theory?
Okay, I think one could be that they use not the correct filters. So I think you should never use filters. We spend a lot of time with some experts from pharmaceutical companies. You should just, I think you should not filter them. But we cannot exclude that the differentiation process or the steps before may alter the sensitivity to retinoic acid. So I think this is a point that should be solved in in the near future. But I would like to stress the point that every atrial EHT model should be very carefully checked if it really resembles findings from human atrium.